This is the last video in the Measuring Microbial Growth and Culturing Methods video series. In this final part of the series, we are going to be talking about culturing viruses. We do not spend much time on measuring viral growth, but we do want to know how we can culture viruses. Culturing is a microbiology term that really just means to grow. So we're trying to figure out how do we grow viruses. The first thing that we need to understand is that there are two different types of viruses. If you remember, we have bacteriophage and animal viruses. Hopefully, you also remember that viruses are non-living because they cannot replicate on their own. Therefore, they need a host cell. Do you remember what the host cell of a bacteriophage is? If you said a prokaryotic cell or a bacterial cell, those are both correct. We're going to go with bacterial cell because bacteria are the host for bacteriophage. However, bacteria are prokaryotic cells. So the method that we are going to be doing here is called plaques. To understand how plaques are working, what we do is we are going to put the bacteriophage into a bacterial sample. So I put my phage into the bacterial culture. Then I'm going to pour it onto a plate. Because there's so much bacteria here, as well as the perfect nutrient requirements for growth, you're going to see bacterial growth across the entire plate. However, what you're also going to see are these holes inside the plate or inside the growth. These holes are called plaques. These plaques are formed because of the bacteriophage. Imagine throwing a rock into a still pond. The rock hits the pond, splashes, and then the ripples go out. And as they go out, they're in this perfect circle and they continue to get bigger. This is exactly what is happening. The initial rock that's hitting the pond is the initial bacteriophage. It's going to infect one bacterium. However, when it bursts out, it's not going to ignore the bacteria around it, but rather it's going to start infecting those. And then when they break out, it'll infect the other ones. And so you're going to get a ripple effect, which is going to form these perfect circles, these plaques on the plate. And this is the only method that we have for bacteriophage. However, for animal viruses, we're going to have three methods. The first method is called tissue culturing. So with tissue culturing, you're going to be growing tissues. Specifically, you're going to be growing eukaryotic tissues because animal viruses, remember, their host is a eukaryotic cell. So I'm going to draw, you have a container, and I'm going to draw cells growing at the bottom. So after you let your cells grow and to start adhering to their environment, you're going to infect them with virus. And the virus is going to spread throughout all the cells. Eventually, once all the cells are dead, there's a liquid media that the cells are resting in. The viruses, because they exit the cells, they will be resting in that liquid media as well. And so therefore, you can now take the media and you can have virus inside that. And so therefore, you're able to grow more virus so you can do more experiments. The next method is the embryonic method. It's commonly referred to as chicken eggs. The reason being is because you're growing the virus in chicken eggs. And the chicken eggs specifically have an embryo in them. So that's my little embryo chicken inside an egg. You're then going to inject this embryo, or this chicken egg, with virus. Because there's ample nutrients and growth of the cells within the chicken egg, because it's in an embryonic state and it's growing rapidly, there's going to be a lot of viral replication occurring. So this is a very great method of growing viruses quickly, and it's also quite cheap. Just so you know, in the embryonic method in chicken eggs, a very common method is to grow influenza. Influenza is grown in chicken eggs, and its vaccine is also grown in chicken eggs. So, if you're allergic to poultry, you actually can't receive the same influenza vaccine as everyone else receives. You have to get a special one. In addition, just as a heads up that you might notice from here on out, if you see that there's a chicken egg shortage in the spring, you're going to see that there is an influenza vaccine shortage during the fall. And that's because they don't have enough chicken eggs to grow the vaccine. The last method is going to be whole animal. So just as it sounds, you are using a whole animal. Commonly, and probably the most common, you're going to be using mice or rats. However, there are all different types of animals you can use. So, I'm going to show that we have a mouse in here. And what you do here is that you inject the whole animal with the virus. Then you just have to feed and water your animal, and the virus will begin to replicate on its own. And this is nice because you're going to be able to not only culture or grow more virus, but you're going to be able to see how the virus acts. So you can see how long is it before the animal gets sick, what are the signs and symptoms, and how long before the animal dies. And so in this method, it's very helpful in observing how the virus acts rather than just growing. But all three of these have the initial method of being able to culture or to grow more viruses. So this is the end of this series, and so I recommend that you be able to understand that in the first series parts, we talked about how there are environmental factors required for bacterial growth, different types of media used to grow bacteria. We talked about how you can therefore see the bacterial growth curve and measure it, and now we talked about how you can culture, or in other words, grow viruses.